record.
Hi, this is Bill Workmeister with Clavis and Harlem River Navy. We're pleased to be joined here today by SciTech, a company operating in the cancer space and repurposing the old J&J drug fenretinide, as well as creating a platform to make hydrophobic molecules, molecules which can't be absorbed or are not soluble within the human body, more soluble. I'm pleased to be joined today by the CEO of the company, Earl Holsapple, as well as Ralph Parchman, another co-founder of the company, and David Schaefer from BDM Collective. Earl, let me hand it over to you first to give a little background on yourself, and then we'll have Ralph and David give backgrounds on themselves as well.
Well, thanks a lot, Bill. It's a lot of fun to be doing this. We don't get the chance to do nice informal dialogues very often. Ralph Parchman and I co-founded the company a couple decades ago, and we've worked together ever since. I came out of industry where I ran razor blade manufacturing companies, food manufacturing and distribution companies, international trade companies, and so on. And we connected at the Karmanos Cancer Institute, where at the time, I think I was a volunteer helping them develop new products. And we've had a great relationship since then in building this business to something we think is extraordinary. So let me have Ralph tell you about his background.
Thank you, Earl. So hello, everybody. I've been involved in developmental therapeutics for cancer since my college days. It's been my passion for decades. And so I've been a guiding principal as I went through my educational courses at Vanderbilt University and studied developmental therapeutics at Johns Hopkins School of Medicine, where I have my doctorate degree. And then went on to do a research fellowship in experimental cancer pathology at University of Colorado with Barry Pierce. And so what I like to do is combine the ideas and concepts of therapeutics and merge them with the biology and pathology concepts I learned in the fellowship to try to tackle the biological foundations of disease. Met Earl, as Earl mentioned, when we were at the Karmanos Cancer Center together in Detroit. I was there for 10 years. As associate professor of internal medicine in the department there at the School of Medicine. And in 2005, I came to join the National Cancer Institute Frederick facility, where I'm currently working on developmental therapeutics problems. As Earl mentioned, so he and I co-founded the company around a patentable discovery that we made, which is to make nanoparticles around fenretinide so that they could be given IV. And fenretinide as a drug can be given full strength to patients and not partial strength, which up to the time of the invention was the only possibility. So I think that's what we're going to discuss later today. But anyways, happy to be here.
Pleasure to have you, Ralph.
Yeah, it's great to be your partner, Ralph. David joined us more recently from stints at Raymond James and other investment banks, and he brings an extraordinary different experience to our table.
Well, thank you, Earl, and thank you, Bill, for the opportunity. So my background is, as Earl alluded to, primarily 30 years in global equity capital markets, working primarily with small and mid-sized companies, helping them, to message themselves to investors quite well, B, work with investors to help them find really great long-term investable opportunities. Some of my greatest client relationships were with folks at T. Rowe Price, at Wellington, at BlackRock, and others. The idea of working with small, early stage companies with very innovative technology has been a passion of mine for years. And given what I thought was a kind of an orphan area within Wall Street, where they were focused on companies that could go public, companies that were already in the public phase. When I decided to retire now almost 2 years ago, this was an area of great interest to me, and I was fortunate to find the partners at BDM who quickly absorbed me into the organization. And we then found SciTech, and a real, what we thought was a great opportunity as they advanced fenretinide and nanofenretinide, into the clinic and subsequently, hopefully, into the marketplace.
Great. Well, thank you for that background, gentlemen. I appreciate it. So I think the first kind of question we'd like to ask, just to kind of set the landscape, because we have so many, investors from so many different industries, is, with so much money and effort that's been spent, looking for a cure for cancer over the last 50 years or 100 years plus, why don't we have a cure for all cancers yet? Why is it so difficult for a country like the United States with so many resources and so many scientists and so many great technologies to fully attack cancer and to solve all cancers?
Ralph sits on the cutting edge of cancer research in his role at the National Cancer Institute. So he's absolutely the right guy to answer that one.
Yep. So it's a fair question. I think the best way to answer it is to counter the question slightly by saying this is not a either or proposition or a yes no proposition. I think if you look over the decades of the 100 years you mentioned, Bill, that there's been ever more advances in cancer treatments, especially treatments for different diseases, especially starting with cancers of the blood with leukemias and some lymphomas. And then more recently, the last 10 years, you've seen tremendous progress in treatment of lung cancers and some melanoma cancers, which used to be fatal and are not anymore with the advent of immunotherapy. And then certainly in the childhood cancers, and very recently, some of the rare cancers where you're seeing FDA approved classes of drugs for use based on a particular mutation that may be found in multiple different cancer types, and they're all susceptible to a particular targeted agent. So although I think cancers in different organs and tissues or different kinds of cancers do have some similarities biologically, in terms of treatment, they have very different susceptibilities. So you have to imagine that each cancer is going to deserve its own forms of treatment to take advantage or to exploit those vulnerabilities. And even within a grouping, for example, breast cancer, there are now molecular subtypes or biological subtypes like triple negative breast cancer that respond very differently to therapy than, say, luminal breast cancer or early stage estrogen receptor positive cancer. So in your question, curing cancer, I think really is curing cancers. And the movement, I think, in the last couple of decades is to realize that and try to match up therapeutic development for particular types of cancers and molecular subtypes of cancers, so that you're not treating everybody with a new drug, you're treating the most appropriate patient population, and that's increasing response rates and improving survival, I think. And you can see that in the cancer statistics, survival statistics recently.
So this is really, this is really a matter of fighting thousands or tens of thousands of genetically and, separately mutated diseases that are all somewhat different.
Could I add an analogy too? So the one good way to understand cancer, I think, is that it's a disease of your own body. It's a disease where fundamentally important biological processes like cell division, cell survival are co-opted and exaggerated, amplified to the point of being exaggerated. It's like a caricature of biology, like a cartoon where someone's large nose is further exaggerated and they become all nose. So it's exactly like that. It's kind of like, by analogy, for example, water is essential for life, but too much results in drowning and way too much results in flooding. So cancer is kind of that overwhelming exaggeration of the biology that's so essential to life. So when you go to treat it, you're trying to treat a cell that has mastered or exaggerated that fundamental biological process. You have to do that in a way that doesn't knock out that fundamental biological process in your healthy tissues, because they also need that process. So I think what that's led to the last couple of decades is people looking for vulnerabilities. So we're moving from chemotherapy to precision oncology. So what are the vulnerabilities? So they're co-opting or exaggerating these normal processes. Why are they doing that? Often mutated genes, like you mentioned, or amplified genes, and looking for susceptibilities because of that, instead of trying to attack it head on and trying to inhibit what they do best. Hopefully that's helpful.
That's very helpful. Thank you, Ralph. So for the next question, with relation to SciTech, could you guys explain just in simple terms your technology, what it does, how it works, and how broadly your platform can be applied
Mhm.
across these many cancers?
I think I've got that one too. Um, yeah, so the technology is the invention of submicroscopic particles that are small enough to be injected into the vein and not cause obstructions of capillaries and the blood vessels. And those small particles are made up of safe, natural substances called phospholipids. And those phospholipids are the components that make up the membranes of your cells in your body. They kind of divide the inside from the outside because when they assemble into these membranes, they don't let water pass through. So it keeps things contained and really defines a cell and a lot of the structures in the cell. So what we figured out was how to take those materials and make these submicroscopic particles. They're nanoparticles because they're even smaller than a cell, much smaller than a cell. And what we found were conditions where we could make not just one membrane with a core of water inside, an empty core filled with water, but actually we could make concentric layers of this membrane like an onion, like layers of an onion. So it's almost a solid particle of multiple layers of membrane. So what that does is provide huge payload capacity, carrying capacity for drug compounds that like to or prefer to exist in the membrane part of the cell instead of the water part of the cell. And fenretinide is one of those molecules. So when we tested these particles for their ability to solubilize fenretinide in a water solution so we could inject it, what we found was that the technology improved its water solubility by 50,000 fold. So
Wow.
it took, for every one molecule of fenretinide that normally would exist in a certain quantity of water, now 50,000 fenretinide molecules could exist in that same quality of water. Now the possibility is there to give full strength fenretinide and get full strength fenretinide doses into the body instead of partial strength doses that everybody else's formulations have been able to achieve. And to do it safely, because the components are materials that are safe. They've been used in previous drug products, so they have a good clinical track record of use.
Interesting. So I guess that was the next question I was going to go into a little bit is, why was fenretinide essentially your first target molecule? What excites you about it? And, what advantages does it have over, other current potential therapies that are in development? I guess one of the answers to why you guys are making this a target for your platform is this 50,000 fold, increase in solubility. But what other characteristics could there be that, have led you to, make fenretinide your first target versus some other molecule that has hydrophobic issues?
Yeah.
Well, we're going to sort of divide that up. So let's start with the history. We were asked while we were at the Karmanos Cancer Institute to solve the problem of delivering this drug in sufficient quantities to achieve the therapeutic effect. Okay. So we chose fenretinide because the NCI chose us, and they were already conducting clinical trials at the Institute, and Ralph was one of the key scientists working on it. And the more drug they gave the patient in a capsule form, it didn't really impact the amount of drug that was absorbed by the body or delivered to the cell. So we were challenged to solve it, first of all. So that's why we chose fenretinide. But then when Ralph and I, after we were asked to do it, we looked at it and said, my gosh, this compound, this anti-cancer agent has such amazing potential based on the hundreds of studies that had already been conducted on it, both preclinical and in human studies as well. And so we said, wow, this should be fun to get into and to try to solve the problem. So that's how we started. And I guess, Dave, you're going to talk about what excited us.
Well, it's really fascinating. One of the reasons that my team really was drawn to SciTech and got us excited, I think, should be the same reasons that others who either have been familiar in the past with fenretinide or have, done a tremendous amount of due diligence in other cancer or oncology therapeutics would see very quickly that there was a stigma that was associated with fenretinide. There was hundreds of or thousands of patients, dozens or hundreds of trials, and they didn't, there was no conclusion that it was an effective drug, that the bioavailability just didn't, materialize. So the result of that was a stigma around the drug that it just, as good as it might have been in the lab, it just was never going to make it in the human body. And along comes the NCI, the studies that they asked Ralph to do, the engineering of the nanoparticle platform that enabled the bioavailability. And all of a sudden, this problem, if you will, scientific problem, was solved. And you then take a look at the fact that there's an incredibly large addressable market for, broadly available cancer-fighting drugs in an environment today where most of the development is in cell therapies and genomic therapies that are heavily personalized and are extremely expensive. So when you can put all that together and you see the opportunity, in fact, also a very short time to commercialization relative to a number of these other therapies, given the data that exists and the guidance provided by the FDA, it was easy for us to get excited. And I think that as investors such as yourself take a look at this asset, I think we're confident that you'll come to the same conclusion.
Thank you, David. So the next question is, I think one that would really interest our investors, but, fenretinide has, so much promise. And, this was originally a, J&J drug, right? The largest pharmaceutical company in the world. So I think an obvious question is, why did, J&J walk away from this? Did they not have the capabilities to deal with these hydrophobic issues themselves? Did they not have the desire? Why did J&J just throw up their hands and say, we're not interested in this asset anymore?
Well, J&J had a multi-decade experience with this drug, starting close to 40 years ago. They were first developing it as a systemically absorbed dermatology drug, for psoriasis and other diseases like that. And it didn't work in those settings. They were delivering the drug in capsules. So then they connected with the National Cancer Institute, and the National Cancer Institute made the case. There's a guy by the name of Michael Sporn, who's now a senior MD at Dartmouth, who worked with them, trying to convince them that this was really potentially a therapeutic. So the next step was they took it into a huge breast cancer prevention study that included more than 3,000 women over a 5-year period, where the drug was shown to be safe and where there was prevention activity. Unfortunately for J&J, tamoxifen was under development at the same time and beat fenretinide to market. So J&J made the decision that they were going to basically hand the rights to the development of the drug over to the National Cancer Institute, because the NCI was very interested in its therapeutic potential. Okay, so they gave a huge supply of inventory to the NCI, and the NCI sponsored decades of additional research to make the case for this drug's use today. So it's not really that J&J abandoned it, it's just that strategically it didn't fit with their strategy that they were working on at the time. However, having said that, Ralph and I were invited to present at Yale University's Cancer Center at an event that was sponsored by JLab's division of Johnson & Johnson. And we were invited to present to the guy that was head of global oncology drug development. We didn't know that's who we were going to talk to initially, but we thought that's fascinating. So we got there, and this guy really liked our technology. And over a 3 or 4 month period, he got back to us and has gotten back to us subsequently, saying, if you get that drug to prove that it works in its new formulation And in the first cancer you tested, and it doesn't really matter, in this case, it's T-cell lymphoma, then we'll re-engage with you is the word to use. I had to laugh at that, so because I said, when the heck did you ever re-engage? When did you ever engage with us? But you know, but the point was, what he really meant was, we'll re-engage with benretinide. You know, so they're interested. They see the value. This is a safe drug with potential applicability across a lot of cancers and other diseases besides cancer, such as viruses. You know, sort of a timely opportunity. So J&J just made a strategic decision that probably was, if they had the ability to look back on it today, they'd maybe regret it. Or if they had the likes of Ralph Parchment on their staff to solve a delivery problem that others couldn't, then they probably wouldn't have abandoned it, wrong word, have handed it over to the NCI. So it's really a function of strategy, probably poor management decisions in hindsight. And today, if we get that same data that J&J is looking for, why there'll be lots more than J&J wanting to get access to our drug.
Mhm.
Bill, could I add something to you on that? That at least
Sure.
in most drug development programs that have a portfolio of compounds and they have to strategize and prioritize resources, for there to be a drug development project that also needs formulation development at the same time, it's almost like saying it needs 2 drug development programs to get to the finish line. So I think this was a drug that's perfect for small business, and our, for example, NIH small business grant funding, because it was innovative, it required an innovative solution. It required a team to focus on it. It wasn't amenable to just routine formulation and go to the clinic. And I think that also would probably frame the decision, I would imagine, on the pharma system.
Great, thank you for that input,
Mhm.
Ralph. So I guess the next question, you know, our investors have put money into a lot of immunotherapy companies over the last couple of years. It's, you know, kind of the fourth paradigm of cancer treatment now after surgery and radiation and, you know, toxic agents like chemotherapy. And I was hoping you could maybe give a brief explanation of what immunotherapy is, you know, enabling the body's own immune system to attack cancer. And then, you know, kind of explain also the immunotherapeutic, you know, effects of your drug, because I know Earl had mentioned to us that there's, you know, also immunotherapeutic component to it.
Sure. All right. So first, I think first part of the question was immunotherapy and how that's possible in the body, how it works. Yeah, it's becoming clear, I think, that for cancers that why don't we focus on cancers that are not caused by viruses? So let's don't talk about HPV cancers, anything like that. But cancers that are not associated, as far as anyone knows, with viruses, the immune system still requires some sort of way to differentiate those cancer cells from the normal cells. And what's become apparent is that that exaggerated biology of cancer results in very extensive cell proliferation and really very poor what's called DNA integrity. So every time your cell divides into 2 cells, ideally you want exact copies of the DNA, the genes in the 2 daughter cells. And what seems to be happening in cancer is they're accumulating defects as those cancer cells divide. And sometimes those defects lead to sequences of proteins on the cells that are presented on the cell surface that are no longer the individual's genetic sequence. They actually are recognized as foreign sequences. So those mutations are probably what the immune system is recognizing and fighting. And what immunotherapy is designed to do is to enhance that that tumor immunity that many people seem to show. And then immune checkpoint inhibitor therapy, things like Keytruda, nivolumab, all those that you're hearing about even on TV now. What they're doing is releasing the brakes. So part of your body's immune system is a safety net, so to speak. So your immune system doesn't turn on your body and cause autoimmune, destructive autoimmunity. And one of those molecules also serves to suppress the immune cell reactivity of cancer cells, because cancer cells, again, co-opted that mechanism to protect themselves from the immune system. So the idea behind these immune checkpoint inhibitor, these monoclonal antibody therapies, is to interfere with the interaction of that mechanism and unleash the full immunity, the full immune reaction against the tumor, which seems to work really well in melanoma, non-small cell lung cancer, some other diseases. It is a very effective therapy and a good strategy, but it's only working in about a third, quarter to 2/5 of the patients. It's not a universal treatment, and that probably has to do with the probability that the genetic changes in your tumor lead to something your immune system recognizes as foreign, and the ability of each individual's immune system, whether they recognize that mutation as foreign or not. It's pretty clear that a mutation in you or me might be recognized as foreign or not foreign, just depending on our own individual immune system. It's not so clear that one mutation is immunogenic in all patients that it occurs in.
Great. That was a great background. So I guess next, can you kind of explain a little bit about how, fenretinide has some immunotherapeutic effects and maybe how that works?
Sure. So based on studies in the literature from several really good scientific labs, the fenretinide seems to have very desirable effects on the immune landscape. So immune landscapes, or what's called the tumor microenvironment, are often suppressive in tumors. So not only that mechanism, that checkpoint mechanism, where a tumor cell suppresses the immune cell next to it to protect itself, but also there are global cytokines, proteins floating in the milieu, and other cell types like neutrophils, macrophages, things like that, that create, and regulatory T cells that create a suppressive microenvironment. So even if the checkpoint inhibitor unleashes the activity of that effector immune cell to kill the tumor cell, it's still going to be suppressed. That immune reaction is still going to be suppressed, nevertheless. So what fenretinide seems to do, it has some properties and some effects on immune cells that would push that microenvironment toward permissive and away from inhibitory. So it's not necessarily going to activate, the immune checkpoint or inhibit the immune checkpoint, but it's going to be facilitated. So it's actually probably going to turn out to be a good combination drug for immunotherapeutics, because it might enhance in a more global manner in a lot of patients that immunotherapy.
So could this be used with checkpoints or cellular therapies or both?
Probably so. We don't have any experimental data on that, and I haven't seen anyone else's data on that. But as we get into clinical development, establish the safety and the activity of fenretinide, obviously its evolution of a therapeutic would be toward combination therapies, adding it to effective regimens, and even moving it more into front line where a lot of immunotherapy is now. And there's every reason to think it's for sure not going to be suppressive of that mechanism. It'll probably be facilitative of that mechanism based on the laboratory data in the literature.
Fabulous. Thank you. So I guess the next question goes away from the science a little bit, but we were just a little curious about the profiles of investors that, SciTech currently has, involved, and, current investors and, investors that are coming in maybe right now. And, what drives these investors to be interested in your company? And, early stage oncology investment is obviously somewhat risky. So, just maybe if you can give us a little idea of who your current investors are and really what's driven their interest in SciTech, we'd appreciate that.
I'll answer the first part of that, and Dave can speak to where we're going. So most of our funding has come, as Ralph had said, from the National Cancer Institute's Small Business Innovative Research Grant Program. So we've been lucky there. We've also raised some money in what they call convertible note rounds. So we issue notes, and those notes are converted into equity at some future date, at some future price. So the majority of the investment dollars we have raised has been using that mechanism, where the convertible note investor gets a significant discount to the valuation that's used in a Series A investment that subsequently follows the convertible note. And they get an interest return. In this case, the coupon is 6%. And, so it's a neat, simple way to put money in without trying to put a value on the business when the business in such a rapid change. And we're putting a
And Earl,
value on it hard.
what did you say the terms were for your convertible note relative to the next round, the discount?
Yeah, so there are two ways. There's a 25% discount to the value that's used by Series A investors, number one, or alternatively, there's a maximum valuation put on the business of $17.5 million. So whichever gives them the bigger discount, that's what they get the benefit of, plus the interest rate on top of that. Okay. So
Okay.
that's how we've raised all of our investment money at this point. We expect to be able to raise some more money that way, as David will talk.
Mhm.
But so I can't sit there. We've certainly talked over the years to venture capitalists, but they're beholden to the drug industry that's primarily focused on the expensive therapies rather than something like ours that's pretty simple and inexpensive. And so we're moving to other fronts, such as your investors, where people want to make a difference, but recognize they have limited knowledge of the industry. So how can you do it in such a way that they're comfortable investing? That's the kind of thing that David can talk to here.
Yep. Thank you, Earl. So I think there are a few things. First of all, in terms of risk elements that you alluded to earlier, Bill, given the data that exists on this asset, going back decades, and the relatively short time to commercialization that we would envision given the FDA's guidance, the risk level associated with this type of oncology investment, we think, is much less than the risk level associated with the typical or even the newer cell therapeutic or genomic products that are being put into the clinic. So I think that's the first thing to address is the relative risk level, which we would argue is less than what most oncology investors are seeing. Secondly, in terms of interest, we're seeing interest from three primary groups. Number one, advocacy groups, those that have a focus on specific types of cancers, in particular T-cell lymphomas, small cell lung, or breast cancer. Second is a number of strategic investors. These would be existing pharmaceutical companies that either have assets that are attacking or staged to attack these particular types of cancer, or have holes in their portfolio where this would be of great benefit to them and their shareholders. They also see the value of the relatively quick time to commercialization and a good return on their capital, their shareholders' capital. The third, is the traditional VC cohort, and to Earl's point earlier, and I think discussions that we've had with you over the last several months is that the VCs are really focused on cool. They're focused on the cutting edge. They're focused on being associated with the most exciting science. And when you find fenretinide is a molecule that's been tested for 50 plus years, and it's been around since the Edsel, kind of, if you want to put it in those terms. It doesn't check those boxes for a lot of investors. They want to be associated with the cutting edge, not not something that you dusted off and took out of somebody's garage. So the investor that we're looking for is somebody who understands general value, can see the differentiation in this asset and its capabilities versus other assets and other opportunities, and then at the same time, recognize that this will not take hundreds of millions or billions of dollars to get to the finish line. This is an asset that we think can be put in front of the FDA for a total of $20 to $25 million from where we are today. And if I could just maybe expand on that a little bit, the first trial, the Series 1A trial, or the Phase 1A trial rather, is likely to consume somewhere in the neighborhood of $3 to $4 million in capital. And upon completion of that trial, we'll have safety data in actual T-cell lymphoma patients, such that getting to a 1B stage will give us the opportunity, to get the drug to a level, or to get enough data from the new entity to put before the FDA. So the potential is there for an approval at that stage, as opposed to going back to full-blown phase 3, very expensive trial. So our sense is it's a fairly.
Maybe Ralph or Earl explain how that works, like how you can pass, just avoid phase 2 and phase 3 altogether.
Yeah, so first off, it starts with disease. So cutaneous T-cell lymphoma, especially the sensory leukemic phase of that disease, is a rare disease. And if you look at the FDA-approved therapeutics on the market, all of them were approved based on open-label, non-randomized phase 2 trials with, between 70 and 130 patients. That's because, number one, there aren't good third line therapies in those patients. And the first and second line therapies in CTCL, they do work, but they don't work for long enough given the age of onset of the disease in the patients. So it's mostly a disease of middle age onset. I can have young patients too, but typically middle age. So you need 20 to 30 years of disease control, and those first couple line therapies don't do it. So what that's done is allowed drugs to get market approval that show activity, response rates in a defined patient population, often that is refractory or has no treatment options. So the FDA has also released a draft guidance encouraging companies like us to talk with them about the design of phase 1B trials and make sure they're sufficiently powered to make those kinds of conclusions if warranted, with the idea that for a rare disease, we don't want to spend a lot of time and have a lot of patients. You certainly wouldn't want a patient in an untreated control arm, a placebo control arm, because they're progressing. They need some sort of treatment. And you also don't have enough, you don't have a comparator treatment, for example, to compare them to. So what, the question is, well, if you don't like this idea, what would you do? What's the better idea? I mean, the idea is to provide the treatment option and evaluate its effectiveness at the same time and use that data to make a decision about whether that drug is approvable or not. And so we were encouraged at our IND meeting. The FDA actually asked us to contact them as soon as we were coming toward the end of 1A, or when we had enough 1A data to what the response rate might be in this disease, with the intention of planning what might be an appropriately powered next stage trial. And we think, based on that draft guidance, the idea would be sort of provisional market approval with post-marketing follow-up, like is happening in a lot of other meetings after phase 3 even.
I was going to, so if I could, so to wrap that up, but we would, look not only for an investor to help us with 1A and 1B, but if there's an investor that would simply get us through 1A, which would be that, let's call it the $4 million ask. I think there would be enough data from that trial to not only, have the rest of the investment need filled fairly quickly, but also, very likely at a significantly higher valuation.
So this kind of goes into my next question, but I was going to ask how much money you guys are raising essentially and how that's tranched out. So you need about 3 or 4 million for the phase 1A, and then how much more for the phase 1B, and how much of that would be remaining on the convertible, and how much that would be in the planned Series A.
So it could be done in 2 different mechanisms, if you will. One would be entirely done in the Series A, and two is you could split out the phase 1A and the phase 1B into let's call it a phase, a Series A1 and a Series A2, or even take the phase 1A and roll that into the convertible note. There's a tremendous amount of flexibility, and
How much in total would it take to fund you all the way through the end of the phase 1B?
that
Well,
would be roughly $20 million.
yeah,
Okay,
and
so we're
that
looking
and
about-
that includes, Bill, a small cell lung cancer addition on top of the T cells. So what we're really doing is getting, as you like to refer to it, two shots on goal, one for solid tumors and one for blood disorders. So, that's a pretty, reasonable price for two cancer indications where, they like to say they're unmet needs. What it really means is there are a lot of people still dying from these diseases and there aren't good alternative therapeutics to treat them. So, and that, by the way, coming back to the question that Ralph was answering, is what's driving these accelerated clinical trial approaches that the FDA is encouraging, is because we need to figure out how to get drugs in patients faster and get more products to market, especially on the rare diseases that Ralph was talking about, which we're targeting initially. And that doesn't mean that's where we're staying, but getting that early approval from the FDA then gives us, a fast process for getting approval in other cancer indications.
Great. So the next question I had, is really one around people. Sal always says that the biggest risk, in any investment is risk in the people and the team itself. So why do you guys feel your team is the right team to develop, this fenretinide drug and either take it to market or take the company itself, to an exit?
I'm going to start with that one. So you've gotten a chance to see two amazing people, David and Ralph. Ralph is one of the most extraordinary scientists in this space that you could find anywhere in the United States. He's really humble and low key, but he's sitting on the forefront of cancer research in his day job. And so we've got amazing expertise and access to at least concepts and ideas of how you develop a product that the average company our size would never have. So we're well suited with people like Ralph on the technology side. We have other people who have similar types of experience. We have a CFO who's a CPA, an MBA, private equity guy, investment banking person with lots of experience in these kinds of deals. We've got David sitting here who brings the experience of an investor who's been on the forefront of small company investments from a public markets point of view. You really can't get any better than that. And I've spent years running different types of businesses that gives me the ability to help tie this together. Plus, there are 9 other people besides the folks you're looking at here who've got all the experience we need to get this into those clinical trials and through them. At that point, we'll see where we go. Okay, if we're going to, the product could be licensed, it could be sold, we could go it on our own, and the team that we will need for that will obviously be different than the team we've got now, and we'll address that as the strategy for going forward makes sense. So if pharmaceutical companies come along and love us like we think they will, well, then our role with the company will probably be short-lived. But if the valuations that they're bringing or the strategic perspectives that they're bringing are short-sighted, then we could go it our own. And we've got guys like David to help us use the public markets to keep going in that sense. So we think we've got the right people, we've got the passion, we've got the understanding, and we've done, Ralph Parchment particularly, he did something that scientists all over the world could not do. They could not solve the delivery problem of a drug that back in the late 90s was touted as the most promising oncology drug under development that year. So, hey, we've got it, and we're different. We've gone at it differently, but we're the team that's made it happen the hard way, not the easy way. We didn't raise the 20 million and then get the approval from the FDA. We got the FDA to approve our product first, and now we're raising the money. So it's different, and it makes some investors uncomfortable, but it shouldn't. We proved that we can do it, and that's really important in having faith in us. I don't know if anybody wants to add to that, but
That's a hard act to follow, Earl.
So in hearing what you're saying, you would expect, an exit after the phase 1A or phase 1B if you got good data from it.
yeah, I guess you don't want to assume anything.
Yeah, I
You
don't
need
think
to
you,
yeah.
I don't think you eliminate any options. I think you have to keep all the options on the table and do what's right for the stakeholders. And, the new investors that join us are going to have, a voice at that table as well.
Fabulous. So just to wrap up with a more global question, where do you guys see, us being in 10 years with regards to managing cancer based on the new technologies that are, currently under development and the things that are expected to come under development in the next couple of years?
Well, there's only one guy at this table that's qualified to answer that. Ralph.
Well, so let's focus on CTCL first. 'Cause that's our focus. So as with many agents that are active in that disease, I think what you're going to see with fenretinide is that it's going to become a backstop for patients who have received the other approved therapies and are starting to progress on those therapies. There's some very important case reports and clinical trial reports of fenretinide in this disease. And what's striking is that patients who have been through the gamut of approved therapies for CTCL and are progressing, there's even a complete responder in the literature was treated in NCI on a trial of a fenretinide that was just sent home after 20 cycles of therapy because she's in CR, maybe cured. I don't know. It didn't follow up, but that's just really unheard of, isn't it? That you have refractory patients, heavily pretreated refractory phase 1 patients that go into complete responses that are durable and are just sent home without evidence of disease after a year of treatment. There's other, and importantly, she had received therapy with an FDA-approved retinoid prior to that. So this is another important point we didn't have time to talk about, but fenretinide is like a retinoid plus. It's got the activity of retinoids, but it also has additional mechanisms of action. There are numerous clinical reports of fenretinide working in patients who are progressing on prior retinoid therapy. And the only way to explain that is fenretinide has an additional unique mechanisms of action, which probably explains its broad clinical activity. So anyway, to answer you back around to your question, 10 years from now, at least with fenretinide, we think it'll be moving more into first line, more into combination, like we were talking about before, and probably moving into the combination to enhance immunotherapy, we hope, both immune checkpoint inhibitor and maybe CAR T cells. It's the same idea. Those CAR T cells have to work in a permissive environment. They can't work in a suppressive environment.
I would add to that two things. I think from what I'm seeing is that cancer or investments in oncology are going to go into two directions. One is to find broad, affordable therapeutics like fenretinide that can work on a variety of cancers and a variety of indications, and in a variety of different populations. And then those that are going to target those conditions that are more complex. And between the two, there should be great advances in the fight against cancer and in oncology, in the science of oncology. So there's plenty of room for both. There needs to be both. And we think we have one of the most elegant solutions, for broadly treating a very wide, number of cancers.
Mm-hmm. Fabulous. Well, I appreciate your gentlemen's time today. This is
Thank you, Bill.
A really, really great interview. Is there anything else that we should know about the company before we wrap up, or is that everything that you guys have to chat about?
Well, the only thing we would like to add is having the opportunity to work with you over the last months has been a great pleasure. You certainly bring a lot of expertise to bear, and you understand this investment space as well as anybody. So thank you for giving us the opportunity to share to share our vision, our passion, and our, and our belief that this is a breakthrough technology, a relatively simple breakthrough technology that could make a big difference. And it could be a lot of fun for investors to participate in something like this that they could never easily afford to invest in the more complex technologies. It would just be too hard, too complex, and you'd have
Too
to depend
expensive.
on the knowledge of a lot of other people. Here, you can look at us and the rest of our team and say, hey, I can have confidence in this, and geez, we can really make a difference. So we can get a good return and make a big difference at the same time, which is really what's, Ralph and I started SciTech with that in mind. We wanted to make a difference, in a big way, and we think we've got the product to do it.
Fabulous. Well, thank you so much, Earl and David and Ralph. We appreciate your time today. I think it was a wonderful conversation.
Thanks, Bill.
Appreciate it, Bill.
Appreciate it.